happy to introduce somebody that I've had the privilege and responsibility of knowing for the last several years, about 10 years roughly, I imagine by now. John Krakow from Johns Hopkins. He is quite the scientist and clinician. Does incredible work, not only in stroke-related rehabilitation, but also does some tremendous work in studying various concepts related to neurovascular coupling and things like that related to neurology. His training from actually has a clinical degree at Columbia and training at both Johns Hopkins as well as Columbia, and that's where I met him about 10 years ago. We've been, I think, good friends ever since. Now he's back at Johns Hopkins as the John C. Malone Professor of Neurology and Neuroscience, and the head of the very well-named LAM lab there. So without further ado, John, welcome.
Thank you. So I have to switch this on. Hold on. It's all a bit complicated. Is that on now? Oh, the button's on the side? Oh, sorry, yes. Is it working now? Is it working? Oh, now it is. Right, sorry. Not knowing how to do AV at Georgia Tech. I'm embarrassed. Okay, it's really lovely to be here. Uncanny weather, very strange, but I'm enjoying it. So I have a number of things to talk about. I'm not exactly sure what I'll skip and what I'll talk about, but we'll see. If people want to ask questions during the talk, it's sometimes better. I don't know why that's ringing. Because you'll forget by the end what you're annoyed about. All right. This is our website. You can find everything that I'm saying at the website. This is Johns Hopkins. This is the old hospital. Is there some way to stop this ringing? I can feel it ringing. No. What about now? Is it ringing?
It's less.
Okay, I'll put it deep into. Is it still ringing? No, it stopped. No, it didn't. I see. Is it ringing now?
Oh, we're good.
That's better, right? Okay.
Hooray.
All right. I didn't want to. So this is the old Hopkins. This is the new one. And the most important thing is please follow us on Twitter. It's the mark of a real lab. All right. So what am I interested in? Well, as Daniel Wolpert likes to say, we're motor chauvinists. We think that the nervous system evolved to move, and that fundamental principles in cognitive neuroscience can be obtained from studying movement, but movement in and of itself, apart from being a model system for cognition in general, is of great interest to all of us. I mean, let's be very honest. You can't go into a bar or pub without there being some kind of sport on the television. I mean, last night I couldn't make it to my hotel because Americans have finally discovered real football. I won't say any more about that, but then you have to ask yourself, everyone was excited, everyone was dressed up, and to watch grown men hit a ball around. Now, why are we so fascinated in watching usually men running around with some kind of sphere which they aim with their feet or with their hands? We're obsessed with it, right? And we don't really know why. Okay, we still don't know the answer. But we also like to watch sport, partake in it. We obviously like to move about ourselves. And then, of course, neurological disease takes away movement. So across this spectrum, we are fascinated scientifically and culturally. And I would argue that we don't have as much communication between these different areas of obsession with movement as we should, because in the world of coaching and in therapy, there's a lot of what Polanyi, the philosopher, called tacit knowledge. People who over time gain experience coaching or treating patients, and then they get annoyed when scientists come along and try and formalize and give verbal expression to this tacit knowledge. So there's a kind of irony that you practice at being good at teaching other people how to practice, right? And so to this day, not only do we not know why people are so fascinated by watching people who are skilled perform, but we don't really know why practice makes us better, right? I mean, that's the ugly secret in 2017, is we do not have a good computational or algorithmic theory of practice. I bring that up because motor learning is really just a version of practice. It's true if you practice your French vocabulary. Anders Ericsson, who basically wrote the book on deliberative practice, started his career studying chess. So all of these domains, whether it's chess, music, or sport, take time to practice, and we don't know why. All right. Why is that relevant here? Well, I'm interested in motor learning, I'm interested in practice, and also because we have this belief that rehabilitating the nervous system after injury is some form of relearning. So let's talk about stroke. So, as you know, about 10-15% of the time, I run the stroke unit at Johns Hopkins and the cases in the neuro ICU. And in fact, I just finished a few days ago in the units. And we see patients devastated with stroke. So, for example, this is an old slide. These are 10 patients. For those of you who aren't familiar, these are axial MRI images, nose forward. This is the back of the brain. And these are diffusion-weighted images. And this is an MRI sequence that allows that detects the trapping of water intracellularly, so it actually picks up early ischemic damage when membranes fail. And so you can pick up, in the last 15 years, we've been able to identify very early on the lesion. And it's very sensitive and it's more sensitive than it is specific. There are other things that make UDWI positive, but it's rare to have a stroke with a real phenotype and be negative on diffusion-weighted imaging. Now, here's the million-dollar question. All these patients have severe hemiplegia. In other words, they have loss of power and control on the side contralateral to where these lesions are. Okay? And the question that has been asked for a long time is, who's going to get better on their own, who isn't? And if we could predict who's going to get better, or who isn't, I should say, could you do anything about it? Unfortunately, and this is sort of comes as a shock as people get older. I mean, just so you have a sense, there's somebody having a stroke in the US every 40 seconds. 800,000 people a year have a stroke. It's the leading cause of neurological disability. And despite its by far swamping, for example, a disease like ALS, and I'm sure all of you remember a few years ago, everyone was pouring buckets of icy water over their heads. People were really, and it raised a lot of money for ALS. 1 in 500 people die from ALS. 1 in 17 die from a stroke. They don't even come close, and yet a lot of people don't even know what a stroke is, and it doesn't get anywhere near the kind of research interest that other areas like Michael J. Fox and Parkinson's disease, you name it. There's something about stroke that is everywhere. Everyone has a family member or a friend who's had one, but people don't know what it is and there's nothing at the current time to be done about it. And what do I mean by that? Well, after you've had your stroke, you spend the first 2 or 3 weeks in a room, and Julie Bernhard, who's a researcher in Australia, published a paper, and it's been backed up by others, that you spend about 85% of your time in a hospital bed not moving, and you spend 60% of your time alone. I noticed a lot of construction here when I was walking around. We love our buildings, right? And at Hopkins, we have a beautiful hospital where everyone is alone in their rooms, modeled more on a hotel than a place where you're actually going to get better, in my view. Now, why is that bad? Well, because it turns out that in that window, there's a rule of brain repair going on, which we discovered back in 2008. And this rule is called proportional recovery. And if you look that up, it's suddenly become all the rage. There's a paper coming out on proportional recovery almost every month. And it's always interesting to me why there are lags between when you discover something and when the rest of the world actually gets it. You know, maybe 8 years isn't such a long time. But why is it so important? Now, what is the rule, first of all? Well, for those of you who aren't familiar, there is a scale called the Fugl-Meyer, which was devised back in the 70s to measure people's ability after stroke. And it was inspired by the fact that people like Brunstrom and others had described that you go what seems to be like a series of stereotypical stages of recovery after a stroke. So at first you're completely paralyzed, you're limp, you have no movement, and then you begin to get fragments of movement back, and those fragments are more inflection than an extension. You can't move the joints independently. They seem to all go together, and I'm sure all of you have seen patients with stroke see this kind of flexed posture like this. And so the goal was to measure the transition between these stages of recovery after stroke. And the insight was that you get stuck at various points along this trajectory. If you're lucky, you pass through all of them back to near normality. If you're unlucky, you get frozen in a particular early stage of that recovery. And what Fugl-Meyer did is he said he's going to come up with a scale to quantify this transition through these stages so that if we actually had a treatment that worked, you could actually measure a change in the exam. And basically the idea was that he wanted to have a number for when you can move out of synergy. So for example, you have to show that you can lift up your arm or without flexing at the elbow. Okay? So it was showing that you could independently control individual joints. Now that scale goes up to 66. 66 is the best for the upper limb. And what we found was that if you have a score, let's say really bad, you're a 10 early after stroke, so you have 56 points you can potentially get back out of a total of 66, that you'll get about 70% of that maximal potential recovery. So you'll get about 70% of 56 back, and then you add that onto the 10 that you already have, and that will tell you where you are 3 months later. All right? And that rule applies really well, except for a subset of patients who are really bad. So about 50% of patients who are near paralyzed will not show this rule, and the other 50% will show it along with everybody else. All right. Now, why is this rule so interesting? Well, it tells you that there's some kind of first-order process of repair going on that is very stereotypical, despite strokes having different locations in patients with different ages. And the tightness of the correlation is depressing, because if you can predict so well where somebody is going to be early on, 3 months later, it means that the intervening treatment is doing nothing. Okay? Now that's a theoretical consequence of the tightness of the correlation. It's since been shown by Kathy Stenier and Winston Biblow in New Zealand that that's indeed the case, that upping the amount of regular therapy that you give doesn't dent this rule. So just so you understand where we are, people are in their rooms, immobile 85% of the time, alone 60% of the time. Most of them are repairing their brain through this process that is independent of anything that's being done for them while they receive homeopathic doses of rehab. All right? And this is another big study out of Holland reproducing the result again. So people are constantly checking to see if this is true. You can also do imaging. This is an older study we did where we basically did multivariate imaging to see whether we could detect a pattern of activation across the brain that was sort of indicative of something systematic going on that would predict where you were in the future. So resting state and some forms of task-evoked functional imaging can be used not to cross-sectionally see where you are, but to actually detect signal that doesn't tell you where you are now, but where you are in the future, and that's what we did here. So again, it lends credence to the idea that there's something going on in the brain that's telling you something about its future potential, its capacity to recover, rather than its current state. The reason why we did this was because we wanted to somehow break the tie in those patients who were severely paralyzed, with severe paralysis, to try and use another modality other than the rule to distinguish those who would show proportional recovery from those who wouldn't. And with sort of basically it failed. And it turns out that the best way to dichotomize those patients who will from those patients who will not show proportional recovery is to do TMS, transcranial magnetic stimulation, and to see whether there's residual corticospinal tract integrity or not. So if you have somebody who's very, very plegic and you stimulate over the primary motor cortex and you can elicit a twitch in the hand, even though they can't voluntarily move that muscle, it tells you that they will actually proportionally recover in the ensuing 3 months. So it's kind of fascinating. You've got the pathway, but you can't use it. But when you do end up using it, you'll get back the 70%. All right. So we're very interested in motor control, and we want to be able to study it in a quantitative manner and subtract out potential confounds like weakness and compensation. So in other words, when you have a stroke, you don't just lose strength, you lose control, and we would like to be able to dissociate those two aspects of the paresis. So to give you an intuitive notion of what I mean, for those of you who see patients, if you go to the bedside of a patient with a moderate degree of hemiparesis and you tell them, squeeze my hand, they'll be able to do a power grip. All right? But then you tell them, open your hand, and they have trouble extending after they've flexed. And another thing they'll have difficulty doing is if you say, just move that one finger. They can't. And what's fascinating and profound is even though they can flex that finger in a power grip, they have trouble using less force and individuating that finger in a precision grip. All right? So there's a whole, I don't have time to talk about it today, fascinating physiology underlying, which is still being investigated, the neural basis for the power grip and the neural basis for the precision grip when you're using the same muscles, but in fact you can exert a large amount of force in unison with the other fingers, but you can't exert less force in just one of those, even though it's the same muscles. It's fascinating. Now, the equivalent of that is to get people to make reaching movements on an air hockey-like table. So all of you, basically, you make the elbow and shoulder be planar, and you make movements like this. All of you will make beautiful straight movements, and those of you who care about motor control know that we obsess about why it is that we make straight movements. It's not trivial, right? I mean, in order to make a straight movement of the hand, you have to perfectly coordinate rotation around the elbow and around the shoulder. Okay, and as you can tell that's difficult because after a stroke you have difficulty making straight, accurate movements because you start having trouble, as I discussed before, coordinating the joints. So you basically become slave to the rotational arcs of your joints when you lose the ability to coordinate them. All right? And so if you use this kind of setup, you can actually quantify this. And the reason why planar is so useful is that there's only one Cartesian solution to one set of joint angles at the elbow and shoulder. Once you go into 3D, it becomes highly redundant. All right? So basically, if you want to look like this, look normal after a stroke, you have to develop normal motor control. Now, it's true, there's a little bit of redundancy in the muscles, which we can talk about, but at the level of joint control, you're going to actually have to move normally and kinematically in order to look like this again. So what we're trying to do then after stroke is how close after stroke can you get to looking like this if your motor control quality is assayed after friction is removed and gravity is removed. So we're looking at control, the arm equivalent of precision grip over strength. Now, if you use assays like that, and there's the setup. Right, so trunk is constrained, air sled, elbow and shoulder basically planar, targets projected on a screen, and then you make these kind of movements. And all of you would make fast out and back movements. I can advance here, can't I? Now, if you do this kind of data analysis, and you look at things, now has everyone here heard of constraint-induced movement therapy? Okay, so this is the home. Steve Wolf is here in Atlanta. And constraint-induced movement therapy, basically the idea is you take the good side and you put it in a sling or in a mitten so you can't use your unaffected side. So you're forced to try and use your affected side, and then it undergoes training for a number of hours a day. So constraint-induced movement therapy is basically the combination of restraint of your good side and training of your affected side. And there are all sorts of variants on this. It was, as I said, it was the basis for the first multi-center, single-blinded trial in rehabilitation called the EXCITE trial. But we were interested, and it worked. Right? There were improvements in functional tasks of everyday life in the patients who underwent this kind of treatment. But what we wanted to see was, did they really get better? Did they really repair their brain? Did they go back to more normal movements, or were they simply compensating with what they had left? And basically what Tomoka Kotaga showed back in 2013, whether you looked at the arm or whether you looked at the wrist, and you can actually quantify these trajectories using something called hierarchical PCA, where you can actually look at the whole time series of the trajectory. And it's nice because you can get a scalar quantity that tells you how close the trajectory that the patient has is to a normal reference trajectory. So you can actually show how they're moving back towards normality. All right? And basically, they don't budge at all. So in other words, constraint-induced movement therapy does not make you better in terms of motor control. Right? What it seems to be doing is teaching you how to compensate on a task-by-task basis. Now, that's fine. I'm not criticizing constraint-induced therapy, but when you talk about plasticity in the brain under the auspices of a treatment like that, I would say it's meaningless. Plasticity in that situation is no different from the plasticity any of us demonstrate when we train on something. So there's been an incredible confusion in the world of rehabilitation and repair, using the word plasticity every time you train somebody on a rehabilitation task, and I think it borders on nonsense. Robotics, so that's another big field. Now, I don't know how many people here are invested in robotics. I just finished writing a book on stroke recovery, and we've done our own robotics studies, and I have to tell you that at the current time, with an exception that I'll talk about importantly in a little while, I have no idea why we do robotics for rehabilitation. Right? If you actually look at the two biggest studies that have been done in robotics, and you read the opening introductions, looking in vain for the conceptual framework leading to the use of the robots, you won't find it. So this is an incredibly good example of where there's an intuitive sense that the technology is cool and that it's so obvious that it somehow should be helpful that one doesn't actually have to do the conceptual biological groundwork. All right? And I'll talk a little bit more about why we fall into these techno traps. All right? There was a word used for it called solutionism, particularly prone in America, where there's going to be a technology that will magically deliver us from our problems. Now, we did a robotic study. 3D robots. This is a Motorica robot. Patients were chronic, and they got 3 times a week for 3 weeks. Very similar to the other studies. And we looked at the quality of people's trajectories before and after. And basically, you got this robust but extremely small improvement in the quality of their motor control. In other words, the opposite kind of result from the constraint-induced movement therapy, where you were simply training to cope with what you have left. Here, you actually seemed to be getting a little bit better at motor control itself, albeit such a small improvement that it would never actually have a functional consequence. All right? But there's a problem. Because what happens here is there are 2 potential questions you could ask. One is, did they really get better in general at motor control, or did you simply teach them on the test, and they basically just used normal motor learning mechanisms to improve a little bit on this particular test, because in 2017, there's no evidence that after a corticospinal tract or motor cortical stroke, motor learning is impaired. Motor control is impaired, not motor learning. Therefore, all of you have an inferior non-dominant arm when it comes to handwriting. So in a way, you could consider that when it comes to the task of handwriting, your non-dominant arm has had a very, very, very mild stroke. Right? And actually, I don't have time to show the data, but if you look at the deficits in the non-dominant arm and you look at them in mild to moderate stroke, they look similar. So let's say that, for the sake of argument, your non-dominant arm has had a very, very mild stroke. Now, you all know that if you start practicing handwriting with your non-dominant arm, you will get better. There have been studies that have done this. Okay. Now, and you will get better and better at handwriting. Would you say that after your handwriting has improved, that I've cured your arm of its non-dominance? Of course not. What I've done is I've used normal motor learning mechanisms on a particular task to improve your non-dominant arm on that task. I have not cured you of non-dominance. Right? But unfortunately, in rehabilitation, when you see task-based improvement, people actually call it recovery. Right? But it's not. Now, the problem is that if you say, well, how do you know that learning is not impaired after stroke? Why don't you study learning? Let's take patients and let's take controls and let's teach them something and look at their learning curves and then say one delted by X and the other delted by Y and do a t-test and go, look, they have a learning problem as well. A lot of studies do that. Completely bogus. You can't actually do it. Now, why not? Why can't you? So here's the kind of idea, and this is important. I'm sorry, but for all of you youngsters in here, you will thank me one day for stopping you from doing this. This also happens in normal motor learning studies. Right? So here's a healthy patient, and this is their performance on the y-axis and their learning. And here's the patient who has much inferior performance because they've had a stroke, right? And they also learn, and they learn, but they don't get as good as the healthy patients. But look, they've done a nice big delta. So delta P stroke is bigger than delta performance healthy. And you go, aha! They learn better, right? And I'm not making this up. I can show you reams of papers that do this, right? But of course, it's meaningless. There are two reasons why it is. One is because you can, this isn't an example of it, but if you decide on a multiplicative measure of the delta, like a percent change, versus an additive measure, I can actually rig it so that you get diametrically opposite results. So that if you chose an additive measure, you'd say the patients were better learners, whereas if you chose a multiplicative one, the controls are better ones. So you're going to have a theory about why you chose a percent change versus an additive change. But that's actually not the deep problem. That's bad enough, right? It's that we don't have the function that this needs to be interpreted, which is the change in the brain required for a given change in performance. Because it could be that for a stroke patient, this delta P needs no more change inside the brain than this smaller delta P needs in the healthy brain. So for example, if you're a top tennis player and you decide you're going to change your serve just by a little bit, it may take you a year. Right? Whereas if you've never played tennis before, it will take you a year and you'll just get better at everything. Backhands, forehands, serves, right? So you've had a much bigger delta. You went from pure rubbish to okay. Whereas the very good player had an incremental change, but had to take as much time training. Right? We don't have this function. And because we don't have this function, we can't interpret this kind of plot. So we can't actually study learning in stroke patients, right? Unless you match baselines. But you can't match baselines. So you can do it if you have two groups of stroke patients. You match their severity, and then you test their learning. All right? So the reason I'm going into this trouble is it's really hard to think logically about behavior and the brain. Okay? So you can imagine someone going, look at this big difference. We're now going to do imaging. We're going to put these deltas into the design matrix, and then we're going to come up with the neural correlate of the better learning in the stroke patient compared to the healthy patients. And there are such papers, functional imaging, brain stimulation, but the whole thing doesn't make any sense from the beginning. All right? So I'm just going to tell you a little bit about a study we did, basically looking at brain imaging, structural and functional, brain stimulation, psychophysics of people recovering after stroke to try and get at the fine-grained truths there. And I'm just going to show you one bit. You've seen this apparatus before. Basically, what I'm going to show you is that we tracked the quality of patients' recovery in the direction of moving towards normality over a year. We just wanted to see how much better do they get through this spontaneous biological recovery that obeys proportional recovery that we've argued isn't impinged upon by rehab. So we just want to see how does your motor control improve spontaneously while you lie in that bedroom alone. All right? And what is fascinating is that basically if you quantify those trajectories on the y-axis and getting down this way is better, here are controls so you get a sense. This is the unaffected side in black and an age-matched control in white. So you can get much better down here. You dramatically improve over the first 4 weeks, a little bit, and then you flatline. Right, so you have an incredibly small window of opportunity to repair the brain after stroke. And this is, the test is very sensitive because, look, you're way down here when you look at controls. So this is not some sort of difficulty in detecting. You just don't improve anymore. All right? Now, remember what I told you. For the first 2 or 3 weeks, you're alone and immobile most of the time. And this is when all the repair is going on, despite you. Bad news. If an alien were to come from outer space and see what the neuroscience is saying in animals and humans and what we actually do for people after brain injury, they'd leave saying we were beyond hope. I think they would do that anyway at this point. But you can do the same thing for the hand. This is stuff done with Jorn Dietrichson, who's up at Western. You basically have an MRI-compatible finger individuation device. I'll tell you about the imaging data, but just to show similarly, in the interest of time, I'll skip through. Again, your hand individuation, your dexterity, improves over the first 3 months, and then it flatlines. These are controls. So you have a little bit longer in the hand to improve compared to the arm, but again, it's a small period of time. All right, so the question you have to ask yourself is, what are we going to do for the hand and arm to try and do better than what these endogenous repair processes are doing? All right? And what we're not going to do is teach compensation, and we know that current rehab seems to be having no impact whatsoever. All right? I'll skip this in the interest of time. This is just, again, showing you what behavioral dissection can do, but we'll have some fun by switching to the mouse. Hopefully this will work. Can you all see that? Yeah, so that's a mouse. I'm a naturalist. And it's incredible that they can do prehension like this. I mean, did you look at that? And this is, they want to use their tongue. They're not allowed to. And this is what they're trying. And then you train them to get better and better at this prehension time. Now, the whole issue of motor control in the rodent as a model for motor control in the human is a vexed one. And I'm happy offline to debate it. Kolb and Weishaw in Canada have made a very good point for musculoskeletal, behavioral, and even neural homology, homology in evolution meaning derived from a common ancestor, for prehension in the rodent and in the primate. It's actually, evolutionarily, rats are closer to us than they are to mice. So there is a distance between mice and humans. But we're going to just train on prehension in the mouse and then give them strokes. So you give them these small strokes over the convexity. This is a cross-section, rather like those MRs, but these are coronal. And basically, this is the motor cortex of the mouse. This is the callosum. They don't have anywhere near as much white matter. This is the putamen. They have very large basal ganglia. So we're about 45% matter in the mouse. It's in the 10% range. And then there's the stroke. And then you train them beforehand on that prehension task. And then you give them the strokes. Efficacy, because it's very difficult, they have to reach, grab the pellet without dropping it in one attempt and smoothly get it into their mouth. Okay? It's hard for them. And then they get worse. And then you wait a week. You let them spend some time in the acute stroke unit, and then you start rehabilitating them for weeks and weeks on the same task. And just like I was saying with respect to chronic stroke, they just don't get back. All right? They move a little bit, and then they just flatline. This is 3 weeks' worth of training on that task. That's much more than actually humans get, but it still isn't enough to get you back. So let's not wait a week. Let's wait a day. Back to normal. Okay, so there's already evidence, and Corbett and others already suggested that there was this window of opportunity that I was already telling you is called proportional recovery, spontaneous recovery. But in addition, the conditions under this spontaneous recovery also seem to be conducive to increasing the gain of your response to training. Okay? And as I said, if this is true in humans, there's an opportunity to really train the hell out of the patient, but they're not. They're just lying there all the time, not doing much. So the alien would just get increasingly horrified. This is the general idea. You delay, you get less bang for your buck with your rehab. Just to tell you how dramatic this is, if it's really true that there's a special window around the time of the stroke where you're going to have enhanced gains to training, then the best way to treat a stroke is to have another one. So basically, we did the same thing. Here's the mouse. We waited a week. It didn't get better. And then we treated it by giving it another stroke. Back to normal.
Where did you do that?
What?
Did you train it?
What do you mean? Why did I what?
Did you start training it right away?
Yes. Right? So in other words, it's being trained. It flatlines, just like we've shown before. Then you give it a stroke, and it's bigger, so it's actually worse, obviously, because you've just added infarct. Right? Unequivocal. Right? Now, obviously, as a neurologist at Hopkins, I'm not advocating giving strokes to your patients after they come into the hospital, right? But nevertheless, it's telling us something, right? And this is just a control to show that it can't be anywhere. So if you do the same thing and you give them a stroke in occipital cortex and you start training them again, they don't move. Okay, so it's a specific effect of inducing ischemia in a motor region. All right. So we reviewed this a couple of years ago. My God, doesn't time fly? But anyway, essentially the point we make is that, and this is a very important point, okay, and it's incredible how, again, it gets missed. Motor learning ability and plasticity is normal in us. In the chronic state, learning is normal, probably, and you basically just do task-specific learning. That's why constraint-induced movement therapy works. You just learn to get a bit better, just like your non-dominant arm does, but you don't repair. But there's something different early after stroke. You have this hyperplasticity where you can actually reverse the deficit. Now, this is just a study we did. It's a little bit complicated. I'm showing it just to show you. This is the coronal section again. Here's the stroke. Here's where we gave the second stroke. This is actually a medial motor area. It is not exactly around the original stroke. You can then image this region that induced the recovery, and you can actually see a reduction in that region of interneuronal markers. So I'm going fast here, but all I'm going to say is that you can find evidence, if you look at interneuronal populations, inhibitory interneurons, of reorganization in brain regions. The sensitive period of responsiveness to training and spontaneous recovery is very similar to the notion of a critical period in development. And there's a lot of work going on right now trying to reopen critical periods. And all I'm saying here is a special case of where ischemia seems to reopen a critical period. And just like the work on reopening of critical period, like people like Takao Hench are doing up at Harvard, there seems to be a change in the populations of inhibitory interneurons associated with reorganization in regions. Now, take-home little message for all of you who believe in map changes, intracortical microstimulation, and all that epiphenomenon, all right? It doesn't seem to be true that you invade regions of the face and leg of motor cortex when your arm region is damaged, and that general idea that maps expand, maps take over. If you actually read the literature carefully, it doesn't seem to be true. Right? It seems to be epiphenomenal overall. Now, I'm not saying that as a definitive final statement, but you should read cautiously what these things mean. And basically what the data seem to suggest to us, and this is a figure in the book that I've done with Tom Carmichael, and it sort of is spoken to by what I just showed you, where you had a distant premotor region that showed reorganizational changes, is that what seems to be necessary for real repair, restitution, is a big behavioral dose, a residual representation for that limb, and a plasticity level that can be either normal or high. So basically, what we saw in the mouse is that you trained them. You saw there was a residual representation in that medial premotor region for the forelimb that was already there, but non-facilitated before the stroke. And then some kind of enhanced plasticity that can actually let the training change the facilitation in that representation. Now, you look at this simple equation that we came up with here, and believe it or not, it can kind of explain all the rodent and monkey literature if you don't allow yourself to be distracted by map changes. Okay? Now, this is just a suggestion. And so you can understand that in a Randy Nudo-like experiment where there's a small infarct in the hand region with residual hand region left, because you've got high-quality residual representation, you don't need that behavior to get restitution. Whereas if the representation in motor cortex is all gone and you have to jump to premotor cortex, then you're going to need a higher behavioral dose and more plasticity to get the same degree of restitution. All right? Okay, so we're not going to give people strokes. So what are we going to do? I've told you that rehab for repair kind of sucks. I've told you that there's an endogenous repair process that seems to be responding to training in the mouse. But how are we going to get what we've just seen in the mouse and in the primate, I haven't talked about the primate, the non-human primate, into the human? Now, one possibility is drugs. So fluoxetine is a drug that seems to induce plasticity, and it actually has been used to try and reopen critical periods. So we used fluoxetine, and what we did is we did, as I did before, but what Steve Zeiler did is they gave fluoxetine early, but waited a week for training. and they got back to normal. So even though before, when you wait, you don't, if you give a drug early and still wait, you get a big improvement. That's good news, right? It means you might be able to extend the period of enhanced responsivity to training with a drug given early. Unfortunately, if you give the fluoxetine late over here, you don't get the benefit. So on our stroke unit at Hopkins, all patients, unless they have a contraindication, are placed on fluoxetine. Not because of just this mouse study. There was a FLAME study in 2011 published in The Lancet that showed a benefit in patients after stroke. It was started on average about 9 days after the stroke and given for 3 months.
Are they also given training immediately?
They just got regular therapy. And it's interesting that if you look at the FLAME study, and I did a back-of-the-envelope calculation on the patients that got fluoxetine, they all were proportional recoverers. So it seemed to shift everybody in the proportional recovery group because their average Flügelmeier was quite low. It's kind of fascinating. It's going to have to be done again, but it was interesting. Yeah. Here it is. Here's the FLAME study. So it started here. They all spontaneously recover, right? But then these ones are riding on top because they were on the fluoxetine. So it seems to be having a potentiating effect on spontaneous recovery. It might not be on the training responsiveness, right? Another way is to do brain stimulation. So I'm not going to go into this in detail. We did a study with Leo Cohen and Pablo Selin a number of years ago where we look at skill learning in healthy people. You basically have to squeeze a force transducer to move a cursor. It's very difficult because the mapping between the transducer and the movement of the cursor along the x-axis is nonlinear. So basically all of you, if you tried it, would be like this. But basically over time you get really good and you go back to 2, back to 3, and it's very hard. And then basically people were stimulated with direct current stimulation over the motor cortex. This was basically looking at TDCS as an instrument to dissect learning, but it turned out that it turned people into super learners. And now there's a company called Halo Neuroscience where you can buy essentially what look like Beats headphones with TDCS in them. Nightmare. But anyway, bottom line is that people who got this training while stimulated with TDCS over M1, remarkably better skill learners than the controls. I mean, it's not subtle, this has been reproduced. So another potential way to potentiate what you're seeing early is to give drugs. Another one is brain stimulation. But there's not a lot of study yet using TDCS early after brain injury. Enrichment is another one. So in other words, I showed you that hospital room where you're alone watching the Weather Channel. Whereas if you put, like Dale Corbett did, rats or mice in with their friends, and they have wheels and ramps and toys, they actually improve on a reaching task, even though they're not trained on that task specifically. That's very profound, right? Some kind of general effect, that not task-specific learning effect. All right? So you should all be asking yourselves now, we get it. What's going to be, if you were given the task, I'm giving it to you now, what would this be for humans? How would you enrich the environment for humans, even other than the despairing mausoleum rehearsal that is the average hospital, right? What would you do? Well, and I'm going to skip this. This is a grad student. This is a video game of skill learning, but I'm not going to go through this now. Let's just answer my question. We won't talk about that either. We'll talk about this, though, before I get to it. You're all thinking about what you would do to enrich the environment, okay? And I told you, enriched environments have the benefit of generalizing. So you're doing all sorts of toy play, and then you do this particular task, and you're good at it. Now, it turns out that if you wear wearables, and this is a study by Conrad, they basically made people wear on a backpack, this is before we had cool small wearables, to track the statistics of everyday life. It turns out that all of us live in these statistical clouds of more vertical than horizontal movements. Right? Look, I'm living in there right now with my pointer. Okay? So the idea then would be, I thought, after knowing this study, was how do we fool people in our human version of an enriched environment to live in this donut of statistical space? Okay? So what would an enriched environment for patients look like? How should we promote playful, non-task-based exploratory behavior? You all still with me? I know it's a lot of material. And the answer is obviously Disney. So what we realized is all along, and I remember when we got at the beginning of my talk, I talked about how we love to watch grown adults kick spheres around. Well, that's also true when we go to Pixar and Disney. And if you look at their early writings, the illusion of life actually meant movements that made the characters look alive. And there are beautiful writings from the 20s and 30s from the early animators who talked about stretch and bounce and exaggerating movements so you felt that these characters were alive. And we love to watch them move. And here, even on the cover of the figure, you see Pinocchio going through his transitions. So we thought we'll go one better, and we'll make you, as a patient, star in the animation. You'll be the character. So obviously, again, you should have immediately understood that you need to be a dolphin. Now, this is a picture of me. It's not just a gratuitous picture of me. It's just National Geographic did a feature about this, and I think they did a nice picture showing the immersion in a robot. I dissed robots, and here's a robot, so we'll talk about why I'm not being a hypocrite. And here's a dolphin. So we spent, with the team, a couple of years at the National Aquarium in Baltimore studying the dolphins, because what we wanted to do was come up with a full-fledged physics engine simulation of dolphins. All right. And basically the idea was that you would have the robot only give weight support, no assistance along the direction of movement, whilst you controlled the intention in a beautiful blue space, the movements of the dolphin. And just think about it. That's true when you move your arm. When I reach for this pointer, I'm not thinking about muscle contractions and joint torques and anything like that. That gets taken care of by my spinal cord. I just want to grab the bloody pointer, right? So that's the same thing we had, the idea behind the dolphin, is the dolphin had complex physics, which you would implicitly learn, but all you wanted to do was to aim the dolphin towards the fish and to avoid the sharks. And we wanted patients to do this within that window that I showed you for motor control, within the first 4 to 5 weeks after stroke, an hour in the morning, an hour in the afternoon, 5 days a week for 3 weeks. They cover about 2 to 3 miles of continuous movements per day with the arm. There's 2 orders of magnitude, at least, than what patients get in regular therapy. All right? It's incomparable. And the therapists who work with us say they had no idea what an hour actually is. Right? The average number of minutes spent moving the arm in a regular rehab session is about 12 minutes, and you make about 20 or 30 movements. It's pointless. Right? But you can't do it without a robot. Your therapist isn't going to move your arm for a mile. I mean, can you imagine? So that's the trial. And I'm going to finish by showing you, if I can, a video of the game. Now, you have to bear with me, because it's always terrifying when you open up a video, because something terrible might show up instead. But I don't think so. All right. So can we turn the lights down? Is there any way we can turn the lights down here? So what you're going to see is a little bit of an introduction, and you'll see the screens, what the patients actually see in this trial, which is ongoing. We've recruited over 20 patients. It's incredible, right? Yes. All right. This is what they see. This is the robot. Yeah, I understand that. I speak dolphin after years. So you have, the first thing you do, there's the exoskeleton, just giving gravity support, no assistant, and you have to catch the fish. And they're moving in that cloud, and then it gets harder. The sharks can eat you. This is a patient playing. So, and I'll tell you something. I've been looking after patients with stroke for a long time. Moisturizer. These are patients in their 60s and 70s who you can't get them out of the chair. I mean, we've had at least a dozen now. They cry, actually. Right? That, it's you're alone in a room. There's music. It's blue. It's almost amniotic. You're basically this intelligent creature. You don't even know what your own arm is doing because we don't care. We're tricking you by where we put the fish and by having the therapist there that you cover that statistical cloud of movements. And the patients say, why isn't this what therapy is like? Where you actually are a character, you embody this animal, it's beautiful, there's music, and it's true, right? And the chronic patients that we've actually piloted this on, and remember, the trial is within this window, they want it. And then you have this difficult decision to make, which is, even if it doesn't work as a treatment, because you've now hit the chronic stage, even though I'm actually not so sure anymore about that, they are having this pleasurable movement feeling that they've been robbed of. Right? They are actually skilled again in another space. And so it's a quandary. But what I would say is you can use neuroscience, logical analysis of behavior, a mission focus, and the technology being slave to the ideas, not the other way around, and actually make a difference. Now, this is a strictly empirical question whether it turn out to be right, but I feel like we're on the right track, and we're going to have to move into this space in an intelligent way to actually not make being neurologically injured the nightmare that it currently is. Thanks very much. Oh, an absolute disgust.
There are one or two questions briefly. There are two slots open for graduate students to get together upstairs, and maybe a few seats up there.
Question here? Yeah, I actually had somewhat of a tangential question. So I read your recent neuron perspectives piece.
Oh, yes. Yes, I walk around thinking I should wear disguise. Yeah, that was an attack on a completely different field.
No, actually, I really liked it, actually, and it's been bouncing around a lot of, I guess, the behavioral neuroscience community, for sure, with a lot of interest. And I'm curious, since you kind of mentioned this, for the students and things like that, do you have any, favorite readings that you would sort of recommend, either for putting together courses or for how we can...
Yeah, I mean, that's a really great question. I mean, you've asked, in my view, the great question, because you said the word read. No one reads anymore. Okay, I'm being serious. I mean, if I were to actually find out how many of you in this room have actually read a book from cover to cover, it would probably be shockingly low, right? Because what we do now is we read the abstracts of PDFs, and we are 1,000 miles wide and 1 millimeter deep. And that's why, actually, we're in this era of technology fetishism and big data and machine learning and all that rubbish. Why? Because, but let me tell you why. No, that's exactly why. My role is to sacrifice myself to the truth, right? No, I tell you what the problem is that we don't actually want to think deeply, read long books, take long walks, slowly work things out. No, we want fast cures. Wouldn't it be great if the technology plus a bit of machine learning could just tell us the answer, right? I don't want to have a difficult hypothesis and design a difficult experiment. Oh, God. I don't want to be like Darwin and have to walk through a forest for years with a notebook and work something out. Mr. Darwin, I know you've applied to our program. Do you do optogenetics? Do you do functional imaging? No, I don't. Thank you, Mr. Darwin. Don't call us, we'll call you. Okay? So what I would say, first of all, and actually there have been studies looking at this, that people who read do more creative science. And I'm talking about novels, let alone books. But after that diatribe, to answer your question a little bit more fairly, in the bibliography of that paper, there's a lot of stuff, and actually we're thinking of compiling some more, and I'm happy to send an email of stuff to read, but read The Evolution of Species, right? Seriously, right? Descent of Man. But somehow, I don't know what happened, but we went from being intellectuals in science to technocrats, right? And then we don't understand everything we do isn't reproduced, isn't plausible. So don't just do something, stand there is what we should do, right? And read more. And there's stuff on our website, and Alex Gomez-Marin, who is one of the authors on that paper, is an unbelievable scholar of obscure psychologists who've hated neuroscience for centuries. And you can go to his website and find it too. But I cannot emphasize enough to those of you who are young enough to still matter to read. Get off Twitter, get off Facebook, stop reading PDFs, and read some books. And I know that's hard, right, because the entire culture is against you. All right? And Milan Kundera has a wonderful novel called Slowness, which I recommend, to talk about how do we get back to slow thinking. You know, just as an aside, my brother is the president of the Santa Fe Institute, and he used to joke with me when he was doing his PhD at Oxford. He said, "Yeah, I've had a really hard day's work wandering around the museum." Right? Because can you imagine saying to your PI, "I think I want to go off to the museum for 3 hours to have a think." Eject. Right? Yeah. Yes.
Very nice presentation. Thank you very much. The last part that you explained about this robotic rehabilitation. I was wondering how would you apply this paradigm to patients with very limited range of motion who may be completely disappointed that they may not
So just so you
to start.
Sure. So just so you know, I mean, the patients in this trial are densely paralyzed. Right? I mean, they're not completely, you're absolutely right, to overcome the inertia of the robot. But what we do is we take away gravity completely. So we're trying to make you weightless. Okay? So you can have Fugl-Myers as low as 6 or 7 out of 66 and be in this trial. It's actually one of the shocking facts that you can be chronically paretic, hand hanging by your side, have gravity removed, and actually still be able to skillfully, visually, guidedly aim your movements. You haven't forgotten how to do the control part, despite weakness not allowing you to lift your arm. It's kind of amazing, right? So you're absolutely right. I'm not sure what to do with people who are paraplegic. I mean, it's a big question for us, what to do with those people. At the current time, you know, stem cells, maybe there's beginning to be the idea that it might be a way to kickstart, but complete plegia is a problem because passively being moved through the space probably isn't beneficial. So I don't know what to do about people who are densely plegic and remain densely plegic. But for the ones who are densely paretic but not completely plegic, who would otherwise not show proportional recovery, I'd like to think that this might get them through further. But that's an empirical question. I do not know the answer. Yeah.
Thanks again so much.